Hi everyone, my name is Cassandra Lima, and today I'll be talking about the study called Spatial Transcriptomics reveals substantial heterogeneity in triple negative breast cancer. It was published in 2004 by Wang and colleagues in Nature Communication. The study looked at how um, gene expression varies across different regions of the TNBC tumors and how that might relate to treatment outcomes. So some background on that. The TNBC or triple negative breast cancer is a type of breast cancer that does not respond to hormonal therapy or HER2 targeted treatments. It's known for being aggressive and tricky to treat. Some previous studies used bulk RNA sequencing to find different TNBC subtypes, but this approach doesn't find, doesn't show where things are happening in the tissue. That's where the transcriptomics comes in and lets us see which genes are active and are exactly where in the tumor they're being expressed. Um, so the main goal of this paper was to better understand the spatial layout of the TNBC tumors, not just what genes are active, but where they're active in relation to things like immune cells and tumor stroma. The researcher wanted us to know how different parts of the tumor contributed to subtype classification, how immune structures like TLS, tertiary lymphoid structures affect outcomes, and if they could use spatial info to predict who might respond to immunotherapy. Here's a quick view of what they did. They used tumor samples from 94 TNBC patients and performed spatial transcriptomics using barcoded slides that captured gene activity in small 100 micron spots. That's about like 200 cells per spot. They also did an IHC staining to mark immune cells and annotated tissue regions manually. They then used computational tools to split the tissues into tumors and stromas um, areas and analyze the gene expression in both. On top of that, they also created 30 gene signatures to identify the TLS um, immune structures that looked like mini lymph nodes and ran a bunch of clustering analysis to the group like to the group to the groups that are similar in tumor regions. Um, One of the biggest takeaways was their 30 gene TLS signatures that was strong predictor of survival and immunotherapy response. As you can see here, patients with high TLS expression did better overall, and that held true not only for their own data, but also in public data sets like the Metabrick and I Spy 2. I think that what's cool about this is that their TLS signature performed better than some of the immune related signatures that already being used. Some more findings. They also have the TNBC tumors that aren't just one subtype, but um, they're not just one subtype, but within a single like tumor strand. Well, not within a single tumor strand, but within a single tumor, there are multiple spatial patterns. They also identified 14 megaclusters of genes, expression patterns, and from those they were able to identify 9 spatial architecture types that were shared across patients. Um, some of these architecture types were linked to better survival and had targetable pathways. For example, one was enriched in immune cells and showed high TLS activities. A major strength is how comprehensive the study was. They looked at gene expression, spatial data, immune cells, and clinical outcomes. But there were some limits too. Some of the limits included the resolution of the spatial data wasn't at a single cell level. So some of the spots had a mix of cell types. So TLSL structures were small and only had a few ST spots covering them. Um, while, and while they validated a lot of the results using bulk data, they didn't have the second spatial data set for comparison. From a clinical perspective, the TLS structure could be super useful and might help doctors figure out which TNBC patients could benefit from immune therapy. And the spatial archetypes might even give more detailed way to classify TNBC, especially for patients who don't um, fit into every molecular stereotype. So overall, this paper showed the spatial transcriptomics and how they can reveal a lot of the um, complexities within the TNBCs. Um, it also goes beyond what we can see with bulk RNA sequence and how they how the organization of a cell in the tumor, especially immune cells, matter for treatment and prognosis. Thank you for listening to my presentation.